Hi, everyone. One of the biggest questions in biology is this: how did we go from relatively simple prokaryotic cells to the much more complex eukaryotic cells that make up plants, animals, fungi, and protists? Eukaryotes are incredibly organized. They have nuclei, organelles, internal membranes, and much larger cell sizes. That level of complexity did not just appear overnight. One of the most important ideas explaining this transition is the theory of endosymbiosis. This theory suggests that some parts of eukaryotic cells were once free living bacteria that entered into a long term partnership with another cell. And if that sounds wild, it absolutely is. Modern eukaryotic cells may actually be the result of ancient microbial cooperation. By the end of this module, you should be able to explain the theory of endosymbiosis and how it accounts for the origin of mitochondria and chloroplasts, and describe the major lines of evidence supporting the endosymbiotic origin of these organelles. Prokaryotes are structurally simple compared with eukaryotes. They lack membrane-bound organelles, and most are relatively small. Despite that simplicity, bacteria are incredibly successful organisms. But eukaryotes are much larger and more complex. They contain specialized internal structures and compartmentalized functions. Scientists wanted to understand how that jump in complexity happened. Did a cell slowly build organelles one piece at a time? Or did some structures come from somewhere else entirely? The theory of endosymbiosis proposes something remarkable: that eukaryotic cells evolve partly through symbiosis between different microorganisms. In other words, complexity may have evolved through cooperation. The basic idea of endosymbiosis is relatively simple. At some point in evolutionary history, one cell engulfed another cell. Normally, the engulfed cell would be digested, but in this case, the engulfed cell survived. Instead of being destroyed, the two organisms formed a mutually beneficial relationship. The host cell provided protection and resources, while the engulfed cell performed useful metabolic functions. Over time, this relationship became permanent. Eventually, the two organisms became so integrated that they effectively functioned as a single cell. According to this theory, some organelles inside eukaryotic cells were once independent bacteria. That means part of your own cells may trace their origins back to ancient microbial partnerships billions of years ago. Mitochondria are thought to have evolved from bacteria. Specifically, scientists think an ancestral eukaryotic cell engulfed an aerobic bacterium, meaning a bacterium that could efficiently use oxygen for respiration. This was incredibly important because aerobic respiration generates much more ATP than simpler metabolic pathways. That extra energy may have helped support larger cell sizes and greater complexity. Over time, the engulfed bacterium became the mitochondrion. Today, mitochondria are found in almost all eukaryotic cells, including your own. So every time your cells produce ATP using mitochondria, you are using structures that may have originated from ancient bacteria. Chloroplasts likely evolved through a process similar to the origin of mitochondria, but importantly, this happened later in evolutionary history. By this point, the ancestral eukaryotic cell already contained mitochondria. In other words, it was already a more complex eukaryotic cell capable of efficient energy production. That cell then engulfed a photosynthetic cyanobacterium. Instead of digesting it, the host cell retained it. Over time, the cyanobacterium evolved into the chloroplast. This gave those eukaryotic cells the ability to perform photosynthesis. Today, chloroplasts are found in algae and plants, where they capture light, energy, and convert it into chemical energy. So, the ability of plants and algae to photosynthesize may actually trace back to a second major endosymbiotic event: an ancient merger between a eukaryotic cell that already had mitochondria and a photosynthetic bacterium. One reason the endosymbiotic theory is so widely accepted is because there is strong evidence supporting it. First, mitochondria and chloroplasts contain their own circular DNA. That is important because circular chromosomes are characteristic of bacteria. Second, they contain bacterial like ribosomes, which differ from the ribosomes found in the rest of the eukaryotic cell. So, to put that in perspective, in eukaryotes, you have linear DNA and you have bigger ribosomes. So we have a 70S prokaryotic ribosome and an 80S eukaryotic ribosome. Third, mitochondria and chloroplasts divide independently through a process similar to bacterial binary fission, while in humans and other eukaryotes, our cells divide through mitosis. They are also similar in size to many bacteria, which further supports the idea that they may have once been free living prokaryotes. And finally, both organelles have double membranes. The double membrane supports the idea that one cell engulfed another. The inner membrane um, and outer membrane are likely that of a gram-negative bacteria that have those double membrane structures that we talked about before. Taken together, this evidence strongly supports the idea that these organelles originated from free-living bacteria. Eukaryotes are evolutionary mosaics. They are a combination of ancient prokaryotic cells that formed a cooperative relationship with other ancient prokaryotic cells that allowed for the development of complex life. And with these two organisms living together, they were forced to compartmentalize some of their metabolic functions to keep things separated so they could work independently. And because of this, these microbial interactions changed the history of Earth. We went from a world of only single-celled prokaryotic organisms to a complex, diverse set of life forms spanning the entire planet. With that, thanks for attending this lecture, and see you at the next one.